So, hello everyone. Welcome to the session on interpretation of next generation sequencing reports. Uh, this is a brief overview of the talk. I'll be speaking in brief about the basics and the different strategies of NGS, which is used clinically, how to read the reports and interpret them using various examples, and understanding the limitations so as to plan further evaluation steps. So, next generation sequencing, if it's shown in a very simplified form here. So, first of all, we take the genomic DNA, fragment it into different millions of pieces, and out of the pieces, we select or capture depending on what we require. Either we are doing an exome or a panel, etc. So that depends on our target, what we are looking for. Once that is captured, then we subject the targets to a massively parallel sequencing where all the selected fragments are sequenced parallelly at simultaneously. So we have thousands of sequences of each original sequence. Then after this is what is the wet lab part of sequencing. Then once we have the sequencing done, the further downstream process is through various bioinformatic algorithms, where first of all, the fragments which are sequenced are aligned to a reference genome. After alignment, the variants at different positions or different loci are called using various algorithms again. And this then we end up with a pool of various variants in different genes, or what is also known as the variant calls. Then out of these thousands of variants, using different filters, we arrive at one or a few disease causing variant, which is supposedly the cause of the phenotype seen in our patient. So this is in very brief the overview of next generation sequencing. Then we have, depending on what our target is and how we are sequencing or testing families, we have various strategies. First of all, there is whole genome sequencing, where we sequence all the genes in the genome, including the coding regions or the exons, the introns of the genes, and the intergenic regions between the different genes. So basically, the whole 3.2 billion base pairs are sequenced in whole genome sequencing. And we have whole exome sequencing, where from the genome, we select the coding regions or exons of all the known genes. So we have discarded the introns and the known coding intergenic regions, and we end up with sequencing only the coding regions or the exons, which is known as whole exome sequencing. Then further, we have clinical exome, or also known by various other names like Mendelium or focused exome, where the only the exon or the coding regions of genes which are known to cause Mendelian disorders are selected. So these are the 5,000-6,000 genes which are known to cause Mendelian disorders. So this is the clinical exome. And still we have NGS panel, where only a certain group of genes of interest are targeted. So we have uh, genes or panels for osteogenesis imperfecta, where osteogenesis imperfecta is a phenotype and caused by some 18, 19 different genes. So we can construct next generation panels constituting the target genes of according to what our interest is. Then, depending on the different strategies or approaches applied in each family, we can either sequence only the proband, which is the singleton sequencing or solo sequencing, which can be exome, genome, etc. It can be duo sequencing, where we can select two affected siblings two affected, similarly affected father and son duo, etc. Then we have trio sequencing where the proband is sequenced along with his or her parents. So these are different strategies, so on and so forth. We can have various permutations and combinations depending on what the family pedigree and availability of the samples. So with this background, we come to the first case here. This is a very simple case showing the particular typical exome report here. So when we receive such a report, the first focus should be on what is the test done here. So we have here clinical exome. So it is not a whole exome, it is a clinical exome which has focused only on genes which are known to cause Mendelian disorders. So this is an individual with a clinical diagnosis of Marfan syndrome, and most of the labs give the summary of the results in a tabulated form as shown here. So here we have the gene along with the transcript, the variant details in the form of the DNA change and the amino acid change here, the zygosity of the variant, the disease associated with the particular variant, and the inheritance pattern, and the classification as per the American College of Medical Genetics and the American Molecular Pathologist Association guidelines. So this is a typical exome report, and most of us tend to, limit our focus to this table and just see whether a pathogenic variant is reported or not. However, I strongly recommend everyone reading exome reports to go further and read the description or the details given subsequently. So the first, the labs give a detail about the variant which is detected. This variant is in this. These details are in the form of the characteristic of the variant, like where is the variant? Is it a missense variant? It is a truncating variant, and what is the predicted effect of the variant? Is it likely to cause a splicing defect? Is it likely to cause a premature truncation of protein, etc.? Then we have information about the various databases, which are mainly two categories: the population databases like the 1000 genome, the nomad, exact, in-house, etc. And we have the disease databases like the AGMD, CleanVar, and various other disease databases. So, the lab will tell whether the variant found in the patient is reported in any of this database or is it a novel variant. Then we have the various in silico prediction tools, which are large in number, and may be consistent or inconsistent in their predictions. Then we have, this is followed by a brief description of the phenotype which is associated with the particular gene that is detected in the individual. Then they have finally the conclusion based on all these evidences of what the conclusion is. Is it is the variant likely causing the phenotype, or is it of uncertain significance, etc. So this is the first part. This is usually followed by the recommendations from the lab, which is mainly in the form of advising Sanger validation of the variant, which may be because the variant has the depth of coverage is low, or else the particular gene has a pseudo gene. So in those cases, validation is recommended. Then most of the times we have the segregation analysis required. So segregation analysis is testing mainly of the parents or affected, unaffected siblings, depending on the family studied, and genetic counseling, of course. So then they will write about the limitations, which is mainly the technical limitations in terms of coverage issues, where all the target regions might not be covered satisfactorily. And basically it tells you what NGS cannot detect. Like they will tell exome sequencing cannot detect large structural variations, triplet repeat disorders, variants in non-coding regions, etc. Then interpretation limitations might also be there in the form of inadequate clinical information, which limits the appropriate interpretation of the results. Then there is test methodology, which gives you the information regarding what platform for sequencing is used, what capture kit is used, the various bioinformatic algorithms used. And here I would like to mention that we should give particular attention to reference genome that is being used, as right now we are in the process of a shift from the older GRCH 37 to 38. So the further interpretation of the variants depends on what reference genome is being used in that particular test. So here two terms were there which need further explanation. One is the coverage. So coverage is nothing but how much part of the target area is actually being sequenced. So it is the width. So if a gene here has 3 exons and all the 3 exons are covered adequately, then it is said to have 100% coverage. Then another term here is depth. So depth is the number of times a particular nucleotide at a loci is being sequenced. So if you will look here, this particular base here, the is sequenced 132 times, and out of the 132 times, 81 is C and G is 51. So giving a ratio of 60 to 40, which is a heterozygous call. So depth is the number of times a particular variant is sequenced, and coverage is the percentage of the gene that is covered. Then we have the ACMG AMP guidelines, which are the recommended guidelines, consensus guidelines used for classifying the variants detected. And these are just guidelines, and various labs use certain modifications as according to the disease being studied. So the particular case of Marfan syndrome here, the variants were satisfying these criteria, and the conclusion was that the variant was a pathogenic variant. So this is how it is done. This basis, let's move on to some other cases where in this particular case, this is a 14-year-old boy with a clinical diagnosis of hereditary motor neuropathy, and the nerve conduction studies showed it to be an axonal type. So the whole exome report of the proband was this, where the lab had reported variants in 3 different genes, all heterozygous variants, and the disorders associated with these genes were two of them were autosomal recessive diseases, and one was an autosomal dominant condition. So the lab also mentioned that in these autosomal recessive conditions, a second relevant variant or a copy number variation was not identified. So despite being autosomal recessive disorders, the lab has reported heterozygous variants in these genes. So this is a very common condition, common situation we all face now, where the lab returns various variants with uncertain significance. So, so now we review and go back to the case to actually see how the reported variants match the phenotype, and also a segregation analysis by testing the parents. So we did a review of the phenotype, and the proband did not have corpus callosum, nor did he have hypomyelination in the MRI. And he also did not show any myasthenia symptoms in the NCS studies. So the segregation, because this was a variant which was showing autosomal dominant inheritance, we decided to go for segregation of this variant first. So this particular variant in tube B4A gene was inherited from the mother, who was clinically asymptomatic. Hence, therefore, it was concluded that this variant is unlikely to be the cause of the phenotype in the proband. So while doing a review of this particular case, we came across uh, 2 reports where de novo variants in the first gene, that is the SLC12A6 gene, caused early onset progressive sensory motor neuropathy. So the OMIM phenotype, which is associated with this particular gene, is an autosomal recessive neurodegenerative condition with childhood onset and mostly associated with agenesis of corpus callosum. However, recently heterozygous variants in this same gene are also reported to cause isolated peripheral neuropathy. So, with this in mind, we went ahead and did the segregation or testing of the parents, and this particular variant was absent in both the parents. That is, it was a de novo variant present only in the proband. So, with this new information, additional information, we reclassified the variant as per the ACMG guidelines, and it came out to be likely pathogenic. So, this final diagnosis was hereditary sensory motor neuropathy, which is caused by a de novo variant in the SLC12A6 gene. So, this case just highlights the importance of doing segregation studies, which are limited in our scenario because of the cost involved. So if we were to do a trio whole exome sequencing in this case, then this would have been sorted out in the first go itself without doing subsequent Sanger sequencing in the parents. So a trio whole exome sequencing is always preferred, but we because of the cost limitations, we tend to go for singleton exomes in many cases. And also the importance of thorough review literature and a reverse phenotyping, where we go back to the patient and do a detailed phenotyping in view of the reported variants. This cannot be more stressed upon. Then this case does not actually show how to read an exome report, but it shows how, whom to test and which test to actually go for, which helps us in appropriate interpretation of the reports to come. So if we so we sometimes come across such families where there are multiple affected families, and the consultant which comes to us for consultation is actually asymptomatic. So this particular individual does not have any symptoms at present, or is healthy at present. However, many of her family members had gastrointestinal cancer, and these two individuals were alive, and the other two had passed away. So, in such cases, if she were to ask for genetic testing for herself and wants to know whether she will be affected or not, then we would not prefer to actually test her on the first go. We would advise her to get one of the affected individuals to come and get tested first. So the preference is always to test the affected individual where where we do not have ambiguity regarding the phenotype. We know that he or she is affected and has to have the mutation. So once we decide whom to test, then which test to actually go for depends on what you are looking for. So in this particular case, we are looking for hereditary cancer syndromes. So we can limit ourselves to actually only testing for those genes in the form of NGS panel, or we can actually go for exomes. The wider test, or the test which we used, it may increase the sensitivity of picking up the cause, but it may also result in identification of more variants of uncertain significance and certain secondary findings, which will be difficult to actually counsel and interpret. So it depends on what we are looking for, and to weigh the pros and cons of each modalities of testing. So these, when we order test or genome-wide testing, it should always be accompanied by proper pre-test counseling, focusing on why we are doing the test, what are the benefits and the limitations of the test, and the alternatives available, whether to test, not test, when to test, etc. Sometimes direct testing of the asymptomatic or pre-symptomatic individuals can be done when we do not have the affected individuals available for testing. However, in such cases, there are various limitations and pitfalls which has to be explained to the individual before offering such testing. So in this family, the test which was done was a hereditary cancer panel. So it was an NGS panel and not an exome, where only the genes related to hereditary cancer were tested. So this particular colorectal cancer mutation was identified in the MLH gene. So once the mutation is identified, the subsequent asymptomatic or pre-symptomatic adult individuals in the family can be tested to look whether they are carry to look whether they carry this particular mutation or not, and subsequently to keep them under surveillance or for further clinical management. Then we have such cases where the reanalysis or the review of the reports actually changed the whole clinical picture. So this was a newborn with epidermolysis bullosa where the parents had brought the sample. The baby was admitted in the NICU at that time, and the pictures and the history were suggestive of epidermolysis bullosa. So epidermolysis bullosa is a very clear phenotype. However, it is genetically heterogeneous where many genes cause the same phenotype, and therefore an exome sequencing was ordered in the form of a clinical exome, and meanwhile the baby died because of complications. So this was the report which was generated by the lab, where they identified a heterozygous pathogenic variant in this particular gene. So this is associated with epidermolysis bullosa simplex. So when we reviewed the report and did our homework, and this particular gene IDGB4 is associated with three types of epidermolysis bullosa in OMIM. So out of this, the autosomal dominant variety is a very mild form of epidermolysis bullosa, which involves only the extremities, and the other are both autosomal recessive conditions. So it was with the clinical condition in the baby, it was very unlikely that this is the autosomal variety. The chances of the autosomal recessive variety being the cause was more likely, and we went and reviewed the case records of the baby and found that the baby actually had surgery for pyloric stenosis. So now the clinical picture was very evident, and therefore we asked the lab to review or reanalyze the report in view of this new information. So the lab reanalyzed and they found another variant in the same gene. This was a canonical splice site variant. So this initial variant was somehow missed in their initial analysis. So subsequently, the parental testing was done, and the parents were found to be carriers of each of these variants, either of these variants, and therefore the compound heterozygosity of the variants was concluded. So the final diagnosis was now epidermolysis bullosa with pyloric atresia because of compound heterozygous variations in the ITGB4 gene. So this has completely changed the clinical picture, since the risk of recurrence and the subsequent reproductive options are different in this case as opposed to the previous report where the lab had reported only one variant in the gene. So this tells us not to take all the reports on face value itself, and undergo a thorough review and do our an appropriate homework. Then sometimes we have such cases where the phenotype is very clear, pointing towards a very specific clinical diagnosis. So this 17-year-old boy had sensory ataxia for the last 3 years, with hearing and visual impairment, dilated cardiomyopathy, and suspecting a diagnosis of a riboflavin transporter defect or a brown violeta von Leer syndrome. Riboflavin high dose was started and he showed good response. So we were very sure that this was the diagnosis, and to further confirm it, the exome report, the exome sequencing of the proband was sent, and this was the variant which was returned. So this is again a heterozygous pathogenic variant or a frame shift variant in the gene causing BVVLS. So, since this is an autosomal recessive condition, we should have another variant in the second allele. So, but analysis did not reveal any second allele, and the coverage of the genes which are known to cause this particular disease, there are two genes, the coverage of these genes was 100%. So, we should pay attention to all these details to determine why we have missed the second variant. So, in such conditions where only one pathogenic variant is identified in an autosomal recessive disorder with a very clear phenotypic match, the possibilities could be that the second mutation is missed. It can be missed because of technical issues like coverage issues where certain parts of the genes might not be covered. In those cases, we may consider patching of the gaps by Sanger sequencing, or the mutation may be missed because it is an intronic mutation in the non-coding region, or it may be a large deletion or structural variation which is not detected by exome sequencing. So alternative methods like CNV analysis from the whole exome data, or a microarray, or MLP for that particular gene might be employed to identify the such second mutations. Or altogether, it may be that we are being misled by the clinical picture, and the patient may actually have a completely different diagnosis. So in that case, we need to again go back and review the phenotype and reanalyze the exome data in view of this information. Then we have such cases where again the clinical diagnosis is very evident, where the baby had global developmental delay with hypotonia and a very high alkaline phosphatase. So such combination of phenotypic features are seen only in hyperphosphatemia with mental retardation. And however, surprisingly, the exome did not show any pathogenic or likely pathogenic variants, and there were no copy number variants in the analysis from the whole exome data. So, what can be the possible causes of such uninformative or negative exome reports? First of all, and very commonly seen, is that phenotype and phenotyping uh, information that we have provided to the lab is incomplete or incorrect. Then the technical causes in terms of sequencing can be a lack of coverage of the causative variant, that is the variant the is not in the targeted region, or it is in the targeted region but it is not sequenced properly. Then it might be because of large indels or structural genomic variants, or it might be a triplet repeat disorder which is not caused by, which is not detected by exome. Or there may be misinterpretation, bioinformatics calling issues. Then the causative variant might be present in the mitochondrial genes, which may or might not be covered by your exome capture kit, or it might be mosaicism, or it might be a new gene disease association which is yet to be reported, and hence a reanalysis or reinterpretation at a later date is always helpful. Or we should also keep in mind existence of phenocopies, that is the phenotype is almost the same as certain disorders but it is caused by non-genetic etiologies. So these are some of the reasons for having an uninformative or a negative exome report. Then in some labs have started reporting, giving additional information, where in such a case with in osteogenesis imperfecta who underwent exome sequencing, the variant for the primary indication, that is osteogenesis imperfecta, is being detected here. This is a homozygous variant in this gene and it is a pathogenic variant. However, in addition to this variant, there are some other variants which are detected which are unrelated to the primary indication. So in this particular case, we have a heterozygous known pathogenic reported variant which is known to cause Brugada syndrome. So this particular child at 2 years does not have any features of Brugada syndrome. However, since we are doing an unbiased or a genome wide analysis and all the genes are being studied at one go, this is such situations arise frequently nowadays. So these are called secondary findings, which are findings unrelated to the phenotype. However, reporting such findings may be useful for the family and for the particular individual. So ACMG has again given guidelines of reporting secondary findings with the latest revision of version 3 now available and covering 73 genes. So, but such reporting of additional results should always be accompanied by proper pre-test and post-test counseling. Some labs may also report carrier status in certain genes, which is not recommended at present. Please keep in mind that this is different from the testing for carrier status in couples, where our primary goal is to identify the carrier status for particular conditions when the proband is not available. So this here we are analyzing the test results of the patient or the proband, and the proband may be a carrier of many variants, but these are not recommended to be reported since it does not have any bearing on his clinical condition. So the proper pre-test and post-test counseling should focus on what the test is being offered, what are the benefits in terms of management issues of the condition, then benefits to the at-risk relatives or the family members. What are the alternate tests available, including doing no test? Then we should give them in a nutshell what results are to be expected, including an uninformative or negative exome and the possibility of finding a variant of uncertain significance. Then secondary findings, limitations, the cost involved, and they should also be explained about the need for subsequent tests like segregation analysis in the family when we are sending only the exome of the proband. So this is a very commonly encountered situation where the family comes with the report and we tell them that we need further testing of the parents to confirm. So this should be explained in the beginning itself, and once we get the report, then we should explain what the report means and the interpretation in terms of again the disease prognosis, treatment, etc. And in terms of VUS, we should also tell them the possibility of change in the interpretation depending on availability of more additional information in the future. So the key points of today's talk were summarized here. So first of all, before offering or before advising a whole exome or an NGS based testing, we should always ask these questions. Is the genomic test indicated in that particular family? And if so, who should be tested and which test should be ordered? This should be always accompanied by a deep phenotyping, and the lab should be provided with maximum possible information, which increases the diagnostic yield of the test. And of course, proper pre-test counseling should be done. Then once we have the report, we should always critically analyze the report and ask whether this particular disorder in the patient is caused by what is reported in the lab report. Then once we have the report, it's always good to go back to the patient and again examine him or her in view of or in light of the new findings or the diagnosis, which is known as reverse phenotyping. And if we have an uninformative test result, we should make a plan of what next to be done, understanding the limitations of the test, a thorough literature review, and there is always a window for reanalysis and reinterpretation of the reports. So thank you very much for your kind, patient listening.